Okay, y'all, welcome back to another lecture in anatomy and physiology. Today we're going over the integument, and I would highly recommend printing out or having your lecture handout up and available for you as we go through so you can kind of follow along. So here are your learning objectives. They correlate very closely with what's in the study guide, and we're just going to go ahead and get going. So we've talked about chemistry, or the chemical level of organization. So atoms make up molecules, molecules make up organelles, organelles make up cells, groups of interacting cells make up tissues. When two or more tissues interact to carry out a common set of functions, they make up an organ. When two or more organs interact to carry out a common set of functions, they make up an organ system. The first of the organ systems that we're going to go over is the integumentary system. And just on an interest note, the integument, which is our skin, is the largest organ in the body, followed by the liver. Now, when you think about what the integumentary system is composed of, we have the skin. The skin is also called the cutaneous membrane. We have hair, which serves a protective and thermoregulation function, meaning it regulates temperature. We have nails, right, which protect the tips of our fingers and tips of our toes and play a role in things like manual dexterity. We have sweat glands. We have oil glands, we have ceremonous glands, which are the wax glands in the ear, and we have mammary glands, which we're not really going to talk about today. We get into the discussion of mammary glands when we go over the reproductive system. So those are the organs that compose the integumentary system, and remember, two or more organs working together to carry out a common set of functions makes up an organ system, and that's why the integumentary system is considered an organ system, and it doesn't work in isolation. It works with all the other organ systems, but it's the one that's most easily observed, and therefore it's the one that's often covered first in an anatomy and physiology course such as this. When we think about the functions of the integumentary system, it is the primary barrier between the external and internal environment, and in that respect, it protects us against things like pathogens, bacteria, viruses, fungi, chemical stresses in the environments like toxins, UV damage from the sun, etc. It's a protective mechanism. It kind of works, it's part of what's considered our innate immune system. It blocks things from getting into our system. If somebody has a burn or a compromise to a significant portion of their integument, one of the biggest things you worry about is invading pathogens because it's such an important barrier to the external environment, and there are so many threats in the external environment. The second thing the integumentary system does is it prevents water loss. So we have waterproofing lipids and waterproofing proteins in the skin that prevent water from just evaporating out. They're not perfect, but they're pretty good. Another concern when people have significant injuries to their integument from something like a burn or from something like road rash, right, abrasion injuries, is that water just starts to evaporate out wherever the integument is compromised, so you also have to pay attention to their hydration status. And you'll notice that burn victims, when they're recovering, will drink everything in the room. You can run liters of fluid on them. It's crazy. The next thing the integumentary system does is in response to UV radiation or to EM energy from the sun, that triggers cells within the integument to convert cholesterol into cholecalciferol, which is really vitamin D. That vitamin D goes to the liver where it turns into 25-hydroxyvitamin D3, and then the kidneys where it turns into 1,25-hydroxyvitamin D3, and it's really important in the absorption of calcium along the GI tract. But the initial point at which vitamin D is being synthesized, meaning cholesterol is being converted into the precursor of active vitamin D, is within the integument. So exposing yourself to a little bit of sun each day is not a terrible idea. Not too much, and it depends on your skin pigmentation, but it's not a terrible idea. Another thing the integumentary system does is it allows for sensory perceptions or sensations. The integument and the nervous system are derived from the same tissue embryologically. So they're very closely connected to one another or linked to one another. So the integumentary system has receptors that pick up touch, pressure, temperature, pain. That information is then transmitted all the way from the integument to the central nervous system where it's processed in the brain. The integumentary system and the nervous system have tight links to one another. Finally, excretion. So excretion of sweat, which plays a role in regulating our temperature. Sebum, which is an oily substance that protects hair. Milk, which is important, for example, for a newborn, the nutritional needs of a newborn. So those are the major organs that make up the integument and the major functions of the integument. Now when we get into the anatomy of the integumentary system, the integument, or the skin, is composed of two major layers. On an exam, I'll ask you the major layer, and I want to make a point of distinguishing the way that questions can be asked. So if I put a bracket around the more superficial layer here and said, identify the major layer indicated by the bracket, that would be the epidermis. The epidermis is the outermost or most superficial layer of skin. If I said, what specific tissue makes up this major layer, that would be keratinized stratified squamous epithelium. So that's a different question when I'm asking you what tissue physically comprises this major layer. So pay attention to that, especially with your handout, because in a moment here, you're going to kind of start following along on the handout and writing in things on that table that you've been given. So the outermost layer of the integument is the epidermis, and then the deep layer of the integument, which is comprised of two different types of connective tissue, which we're going to discuss in a moment, is called the dermis. So identify the major layer, epidermis and dermis. And this kind of goes into the handout that you're going through as you follow along with me. So we have the skin. Now the skin has the epidermis and the dermis. The epidermis in thick skin is composed of 5 layers, and in thin skin is composed of 4 layers. The tissue type present here would be keratinized stratified squamous epithelium, and then we're going to talk about cells and their function and other structures and their function. We're going to go over thin skin first, starting deepest to superficial. So this is the deepest layer right here. It's the same in thin and thick skin. It's the stratum basale. And then we're going to work our way superficial. We're going to work our way up. These are sublayers of the integument, or specific layers of the epidermis. They're sublayers of the epidermis. The dermis is then composed of 2 different layers, each of which is made of a different type of tissue, each of which has structures and cells within that region. Now, the hypodermis is not actually part of the skin. It's not considered part of the integument. So the hypodermis is actually part of what's called the subcutaneous fascia. It's just a network of connective tissue that separates the overlying integument from the underlying muscles. It's very blood vessel rich because it's composed predominantly of adipose tissue, so that's the tissue type present. Don't worry about cells and their function and other structures in their function. Hypodermis, deep to the dermis, composed of adipose tissue, not actually part of the integument. It separates the integument from the underlying musculature. So what we're going to go over first are the specific layers in both thin and thick skin, and we're going to talk about the difference in the epidermis between thin and thick skin. And then we are going to talk about things like cells and their functions, and we're going to go through it like that. So it gives you kind of an idea of what you're doing on the handout here. Your next image on your handout is actually showing you an image of the epidermis in which I've delineated the different layers. So I would label those layers because that's probably going to come up on something like a lecture exam with respect to a short answer question. So, specific layers of the epidermis, we're going to start with thin skin. You find thin skin everywhere on the body, with the exception of the palms of the hands and the soles of the feet. So thin skin is found everywhere on the body with the exception of the palms of the hands and the soles of the feet. Thin skin is composed of 4 layers. So starting with the deepest layer, we have what's called the stratum basale. The stratum basale is a layer of stem cells that's constantly dividing to replace more superficial cell layers. So these stem cells in the stratum basale are constantly dividing to replace more superficial cell layers as they slough off. Every time you touch your skin like that, you slough off literally millions of cells. So you constantly have to be replacing those cells. In fact, your epidermis turns over or replaces itself every 28 to 40 days. So this stratum basale, this layer of stem cells, is really important because it's replacing those more superficial cell layers. Now, some cell types found here Keratinocytes, right? So when you look, keratinocytes are actually just skin cells. So 95% of the epidermis is composed of keratinocytes. Keratinocytes are skin cells. They're called keratinocytes because skin cells crank out a structural protein called keratin. So another type of cell you find embedded within the stratum basale, these large cells with these projections, are called melanocytes. And melanocytes are responsible for synthesizing a pigment called melanin. Melanin that gets deposited in our keratinocytes where it protects the nuclei from UV radiation or UV damage, right? So it's skin pigmentation. There's a skin pigment producing cells. If you have lighter skin tones and you're in the sun, you can actually upregulate the activity of melanocytes, and that's what a suntan is. So as we work our way up, the next layer is what's called the stratum spinosum. It can be anywhere from 10 to 15 cell layers thick. The number is irrelevant. What I really care about is that you know that these are metabolically active or living cells in the stratum spinosum. So they still have nuclei. You'll notice they still have nuclei. They're still living. And what these cells are doing is cranking out both keratin and little lipid granules, right? Little fat granules. And the keratin is a structural protein. It also plays a role in preventing water loss. The lipid granules play an important role in the prevention of water loss. So this is a metabolically active layer in which the cells are cranking out keratin and these lipid granules. Within the stratum spinosum, you have a cell type, and this is where you would write that for the thin skin and thick skin. This cell type is called the Langerhan cells. Langerhan cells are immune cells that detect immune threats and then trigger the immune system. So if you ever have a reaction in your skin, like an allergic reaction in your skin, it's the Langerhan cells that, for example, picked up that poison ivy and triggered an immune response. They're immune cells because we're exposed to so many threats. Now, you have the stratum spinosum, which is metabolically active living cells, but the cells eventually undergo a process called apoptosis, A-P-O-P-T-O-S-I-S. And in apoptosis, cells undergo programmed cell death. So the cells die. And when the cells die, they lose some of their water content and they pick up stain a little bit better. So in the stratum granulosum, cells undergo programmed cell death or apoptosis. They die. And you can see all of those lipid granules and all of that stuff becomes clearer under a microscope because they lose water. The cell's content becomes more concentrated and it picks up stain a little bit better. So that dark staining grainy area is the stratum granulosum. Those cells are no longer living. Those are dead cells. They've undergone the process of apoptosis. Those keratin-rich cells will then compact and they'll form the most superficial layer called the stratum corneum. Notice that the cells in the stratum corneum, which is the thickest and the toughest layer, no longer have nuclei. These are dead cells, and when you look at somebody, you're looking at their stratum corneum. That's what you're physically looking at. When you touch somebody, you're touching their stratum corneum. So they're just sacks of keratin proteins and these little lipids, and it's a tough layer. These cells are latched together. They're full of keratin, which is a structural protein. Really important layer, very tough layer. And if you expose your skin to stress, you can develop calluses. That's actually just a buildup in the stratum corneum. So those are the 4 layers of thin skin, which you find everywhere on the body, with the exception of the palms of the hands and the soles of the feet. Thick skin, on the other hand, differs from thin skin in the fact that you find it on the palms of the hands and the soles of the feet. Why would you find thick skin, right? Physically thicker skin on the palms of the hands and the soles of the feet? It's because the palms and the hands and the soles of the feet are subject to a lot of mechanical stress, and you need tough stuff there. We're constantly interacting with the world with our hands. We're stepping on our feet, applying hundreds of pounds of pressure. We don't want the skin there to tear, so that skin is significantly thicker than all of the other skin in the rest of our body. Now, thick skin differs from thin skin in the fact that it is actually thicker. It's significantly thicker. And it has an extra layer that thin skin doesn't. It's a layer unique to thick skin. So you have the stratum basale, the rapidly dividing stem cells, the stratum spinosum, which is the layer of metabolically active living cells that are cranking out keratin and lipid granules for waterproofing and protection. You have the stratum granulosum, where the cells undergo the process of apoptosis or programmed cell death. In thick skin, after those cells undergo apoptosis or programmed cell death, because there's such a thick stratum corneum, those cells compress to form a kind of a very structurally tough layer, a resilient layer called the stratum lucidum. And the stratum lucidum is named that because it's kind of translucent, but what that is just a layer of compressed cells that provides a lot of extra protection to the underlying skin layers and prevents the skin from tearing. So you have the stratum lucidum, and then the stratum corneum is significantly thicker in thick skin than it is in thin skin. Notice no nuclei, because those cells are dead. Why do you have thick skin on the palms of the hands and the soles of the feet? You need tough stuff, subject to a lot of mechanical and chemical forces like washing your hands, the sweat on your feet, etc. You don't want that skin tearing, so you find thick skin in those regions for that reason. One of the things that people with darker skin complexions notice is that the skin on the palms of their hands and the soles of their feet is a little bit lighter than the skin on the rest of their body. That's because melanocytes exist within the stratum basale, and because thick skin is so much thicker, that extra layer, the stratum lucidum, and that thicker stratum corneum actually mask that pigmentation in those deeper skin layers, so you can't see that pigmentation quite as well. If you were to kind of peel off those skin layers, the skin pigmentation would become darker. It's just masked. Now, the dermis. So now we go down one, and we go, the dermis is composed of two layers. We have the papillary layer of the dermis, which is the most superficial. It interacts with the epidermis, and the epidermis latches down to the papillary layer of the dermis. This is definitely thin skin. In fact, that's like skin from the lip. It's super thin skin. This is the thinnest skin you have is on your lip, and that's why, you know, the lip busts so easily, or when you can see the blood vessels in the underlying connective tissue more readily, which is what makes your lips red, because the skin is so thin. So you have thin skin of the epidermis. Then you have the two layers of the dermis. The papillary layer is the first layer of the dermis. It's made of areolar connective tissue. That areolar connective tissue is what gives your skin a lot of those elastic properties and allows the skin to kind of bounce back to its original position once you distort it a bit, like you grab it or you pinch it. The deeper layer of skin, which is much thicker, or dermis, skin in the dermis, which is much thicker and much tougher, is called the reticular layer of the dermis. The reticular layer of the dermis is composed of dense irregular connective tissue. You can see those collagen fibers running in every which direction. That withstands pull forces from multiple directions without tearing unless the pull force is adequate to tear. And dense irregular connective tissue is like leather. So when you think of the reticular layer of the dermis, I want you thinking like leather. Why is it such an important layer? That's where most of the structural integrity of the skin comes from, its ability to withstand tear, etc. Now, what we've just been through is we've been through thick skin, thin skin. We've talked about the tissue type present, and I highlighted any cells and their respective function. Then we went through the layers of the dermis: papillary layer, reticular layer, papillary layer, reticular layer, areolar connective tissue, dense irregular connective tissue, and we haven't gotten into talking about cell types and their functions yet. So now, cell types of the epidermis that you need to know. Keratinocytes are just keratin-producing cells of the skin. They're skin cells. They just crank out keratin. 95% of the epidermis is composed of keratinocytes. That's one type of cell you need to know. It's a fancy word for a skin cell. Melanocytes are melanin-producing cells found in the stratum basale, peppered in the stratum basale. They crank out a pigment called melanin. Melanin protects from UV radiation. It protects from the sun. It's sunblock. And it has a lot of other interesting properties that we won't get into. Langerhan cells are immune cells. They trigger the immune system when they come into contact with something that the cells themselves interpret to be a threat, like a bacteria or a virus or whatever it is. Merkel cells are little sensory cells. We don't talk about them that much, but they're little sensory cells or fine sensory cells within the stratum basale that interact with neurons and trigger essentially sensory perception that gets sent to the brain. So when you think about keratinocytes, Keratinocytes can be damaged in response to UV radiation, right? You want to, if you're in the sun, you want to limit your time in the sun if you have lighter skin complexions, or wear something to protect your skin from the sun. Don't just go out there after a long winter and suntan for 8 hours, because that can damage those cells. And the cells that you're kind of most worried about being damaged are the melanocytes and the cells in the stratum basale, because if you damage those, that can trigger things like cancerous changes. So erythema solare is just a fancy word for a sunburn. Now, we won't talk too much about premalignant changes, but one of the things you see, one of the most common cancers that people will develop throughout the course of their life is actually what's called a basal cell carcinoma. And in basal cell carcinomas, what happens is the cells in the stratum basale suffer a mutation to their genes, so a DNA mutation. You change the code in the DNA, and that mutation can lead to a situation in which those cells start to divide uncontrollably. When they start to divide uncontrollably, they can produce a little cancerous tissue. Cancerous cells are just cells that are constantly in mitosis. So the cancerous tissue can grow, if it's stemming from the cells in the stratum basale dividing uncontrollably, into what's called a basal cell carcinoma. This is not that dangerous. Many people will develop these throughout the course of their life. It has a very, very high survival rate. It's usually just a removal, one and done, and you're done. Now, melanocytes. Melanocytes are peppered in here through the stratum basale. I'm not going to ask you to identify them histologically, but what they do is they crank out a pigment called melanin, and melanin is what gives us our skin color, or it plays an important role. It's an important pigment determining skin tone or skin color. Melanin protects all of these cells, these living cells in the stratum basale and in the stratum spinosum, right? That melanin clusters into those cells. It gets cranked into the keratinocytes, and it essentially wraps around the nuclei, and it prevents the DNA from damage from the sun. So it absorbs the energy from the sun, and it prevents that energy from damaging DNA. That's the same thing sunblock does. So skin color, the activity of those melanocytes, is what's called a polygenetic inheritance trait. There are many, many genes associated with it, and you can have people who express those genes on one side of the spectrum and on the other side of the spectrum. Now, diseases that you might hear about that impact melanocytes, there's vitiligo. So vitiligo, there's impetigo, which is a bacterial infection, and vitiligo, which is an autoimmune disease that actually destroys melanocytes. And when those melanocytes are destroyed, they can't crank out melanin, and it actually produces this kind of patchy or blotchy skin appearance. Vitiligo is a little bit harder to treat than impetigo. Impetigo is a bacteria that destroys melanocytes, and you get this same splotchy appearance. In that case, you can typically eradicate that bacteria with an antibiotic, and the skin will pick back up where it left off. With vitiligo, which is an autoimmune disease, that means your body's own immune system is targeting those melanocytes, and that's a little bit trickier to treat. Melasma is an outbreak of freckles. It's very common during pregnancy because many of the pregnancy hormones can actually upregulate the activity of melanocytes, and you can develop these freckles, which can take a long time to go away. It's nothing to be concerned about from a health perspective. Some people don't like it aesthetically. Now, melanocytes can also suffer damage from things like UV radiation. And they can have mutations, and when melanocytes, those cells have mutations in their DNA, that can influence the way they divide. When a cancer is stemming from a melanocyte, that's a much more dangerous cancer than a basal cell carcinoma. It actually produces a cancer called a melanoma. Melanomas are a lot more dangerous because not only do they grow out, but they grow in. And when they're growing into those blood vessel networks, they have a much higher likelihood of metastasizing or spreading because some of those cancerous cells can slough off. And because of that, they're just much more dangerous. These actually have a mortality rate associated with them where basal cell carcinomas don't. Well, they have a much higher survival rate, I'll put it like that. So melanomas are dangerous. So you have Your melanocytes, they start dividing uncontrollably. They're cranking out melanin at, they're not regulating their cell cycle or their activity very well anymore. So when you look at a melanoma, there's actually the ABCDs. If you sprout a new, let's say, mole, what you think is a mole or freckle, but it's large, right? And it's asymmetrical, meaning it's not kind of circular shaped. That's the A. The B is the border. Is it a rough border? The C is the coloration. You see different colors. Most moles or benign moles have homogeneous colorization, meaning that they're brown throughout, not like purple brown, dark purple, a bunch of different colors. And if the diameter is greater than 5 millimeters, then yeah, you should get it checked out because melanomas can be really, really dangerous. So that's a melanoma. That stems from inappropriate division of melanocytes. So I'll tell you one thing. I had a friend in Australia who had a melanoma on his nose, right? Just a little melanoma. It looked like a dot. The melanoma had grown into his nose, into the cartilage in his nose, and in order to treat it, they had to go blah, right? And essentially cut his nose off. That's a really aesthetically not a very pleasant thing, but he was saving his life. So they had to literally take essentially most of his nose off. If you're out in the sun, right, a lot, one of the worst places you could possibly develop a melanoma is on your ear or your nose because they're just going to take that off and there's not a lot of good reconstructive surgery. That's dangerous, just on a by the by. Now, another pigment that influences the color of skin in both individuals with lighter skin tones and darker skin tones, but you see it more prominently in individuals with lighter skin tones, is hemoglobin. Even though hemoglobin is the protein found in red blood cells that transports oxygen, you can see it in the skin. In fact, whenever you squeeze somebody's finger and you see it go from being white to red again, that's called capillary reperfusion. Cardiologists use it as a marker of your cardiovascular health all the time, meaning that if it stays white for a prolonged period of time, your heart isn't working very well, your cardiovascular system isn't getting blood to your extremities very effectively. And you see it go from being white to red or kind of pinkish. That's because the blood vessel networks underneath, right, the skin that feed into the skin, get clamped down, so you stop blood flow, and when you release it, blood flow is reestablished, and you can see the hemoglobin, that red pigment, kind of, right, in the blood rush back in. So hemoglobin is a pigment that influences skin color too. Now, Langerhan cells are immune cells. If you've ever had something like a skin prick test to determine what you're sensitive to in the environment, like an allergy test, what they're doing is they're trying to trigger Langerhan cells to see whether you have an acute allergic reaction. So the first thing they inject into your skin is something that they know for a fact is going to trigger an allergic reaction because they need a control. The thing that they inject into the skin that they know for a fact is going to trigger an allergic reaction is histamine. So you inject that histamine into the skin and you go, okay, allergic reaction. We know it's going to be an allergic reaction. Then you compare everything to that. This person actually has allergies to a wide array of substances. It's not normal to see that. This person probably has what's called multiple chemical sensitivity. But when you get that pinprick in the back, the cells that they're trying to activate are those immune cells in your skin called Langerhan cells. Now, skin structures within the dermis that you need to know. Here are three. 1, 2, 3. We're going to go through those one by one. If you're continuing to go through your handout, you'll notice that you have this exact same image, so we're going to reference back to this image quite frequently. Now, in the papillary layer of the dermis, which is composed of areolar connective tissue, all of these projections, these kind of finger-like or nipple-shaped structures right here, are called dermal papillae. In fact, papillae means nipple-shaped structure. And you have millions of these. And these dermal papillae, right, insert into these epidermal ridges, and that's what locks the epidermis down to the dermis. What it does is it functions almost like Velcro, and it increases the amount of surface area the epidermis has to lock down to the underlying dermis. It also increases the amount of surface area available for things like the exchange of gases and nutrients between the blood vessel networks in the dermis and the avascular cells in the epidermis. So the epidermis, right, remember that epithelial tissue has no blood vessels, gets all of its nutrients and gases and gets rid of all of its waste in the blood vessel networks of the underlying dermis. So these nipple-shaped structures are called dermal papillae. They're genetically unique in each person, so when they project up into the epidermal ridge pattern is unique for each individual, and you can see that really clearly on thick skin with things like your fingerprints. Capillary loops. Blood capillaries are the most abundant and most important blood vessel within the cardiovascular system. You have billions of them. So cardiovascular 101. Heart beats. Blood moves away from the heart in large vessels called arteries. Arteries branch and branch and branch again and become very, very small. Arteries carry that oxygen-rich blood in the systemic circulation to the point of exchange called a capillary. Red vessels indicate oxygen-rich, blue vessels indicate oxygen-poor. So at capillaries, the reason that they're the most important is this is the point of exchange. You deliver that oxygen-rich, nutrient-rich blood, right, or oxygen and nutrients to the cells in the stratum basale, and the stratum basale gets rid of all the CO2 and metabolic waste, which then leave and ultimately become part of a vein. That vein then brings that blood back to the heart, and then the carbon dioxide is eliminated when you exhale in your pulmonary circulation, and that's kind of a quick overview of what's happening there. So when you see a blood vessel go from being red to blue, the only blood vessel that can do that is a capillary. As a consequence of that, you know you're looking at a very small blood vessel, and you know you're looking at a point of exchange. Capillary loops are there to facilitate exchange between the connective tissue and the epidermis, meaning that they deliver oxygen and nutrients to the overlining epidermis, and that's what allows those cells in the stratum basale and those melanocytes, all of those cells, to get their nutrients and their oxygen so they can be metabolically active. Now, the subpapillary vascular plexus is this network of blood vessels that feeds into those capillaries. This is the delineation point or the border between the papillary layer and the reticular layer of the dermis. And it's a little bit more easily seen on that model. So this is the model that you need to know. Over here are dermal papillae. All of these guys are capillary loops. Think about a question like identify the structure. And then this right here, this network is the subpapillary vascular plexus. Now you can see a lot of things really well on this skin model. This is the integument model used in just about every classroom I've ever been in when you have an integument section. So you have thin skin, stratum basale, stratum spinosum, stratum granulosum, stratum corneum. You have thick skin, stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, stratum corneum. Strata means layers, so you can actually see it's physically thicker. You can also see that there's no hair growing out of thick skin, despite what people may have told you during your adolescence, right? So you have those structures that we've just talked about. So it's kind of important to note, and here we'd be seeing a transition between the thin skin of the entirety of the body and the thick skin that you only find on the palms of the hands and the soles of the feet. Think about this model. We're going to come back to that model quite a bit. Now, sensory receptors within the skin that you need to know. Meissner's corpuscles, aka tactile corpuscles, are responsible for detecting light touch. Pacinian corpuscles, aka lamellated corpuscles, are responsible for detecting deep pressure. And free nerve endings are responsible for detecting pain and temperature. So let's go ahead and look at a Meissner's corpuscle. A corpuscle is a fluid-filled sac with dendrites or nerve endings in it, right? Somebody touches you lightly, like strokes the side of your face, or something touches you really lightly. That's going to distort the membrane of this fluid-filled sac, which is going to trigger an electrical signal in the nerve endings within, and that electrical signal is going to travel along the axon of a neuron all the way along a nerve to your central nervous system and up to your brain where the conscious perception of touch is taking place. So we talk about these a lot when we get into the nervous system as well, because the nervous system and the integumentary system are so tightly linked, not metaphysically or philosophically, but physically. Pacinian corpuscles, aka lamellated corpuscles, look kind of like sliced onions. They have layers to them. And because they're much deeper in the integument, or embedded much deeper in the integument, it takes a lot of force to physically distort a Pacinian corpuscle, and therefore they detect deep pressure. So if I said somebody's getting a deep pressure Swedish massage, or deep pressure is applied to the skin in some way, then we're talking about the activation or the disproportionate activation of Pacinian corpuscles, and their location within the integument kind of tells you about their relative functions. Light touch, deep pressure. Now, we don't have any free nerve endings on this particular image, but free nerve endings extend up into the epidermis, where they're responsible for detecting pain and temperature. So if somebody spilt a cup of hot coffee on you, or you got a paper cut, or whatever it was. Now, histologically, here we have the epidermis, here we have a dermal papillae, right? And that epidermis is being locked down to the dermis by these dermal papillae. And within those dermal papillae, we have these Meissner's corpuscles. And I think they look kind of like cotton candy. In those Meissner's corpuscles, there are free nerve endings or dendritic endings. And if you distort those Meissner's corpuscles, which are capable of being distorted with light touch, it will trigger an electrical signal that will then travel along a nerve to your spinal cord, and then up from your spinal cord to your brain, which is where you'll perceive the light touch. They're also called tactile corpuscles. Pacinian or lamellated corpuscles, on the other hand, lamella means sheet of tissue. They look like sliced onions, and they're deep. So here's the epidermis. Here are your dermal papillae, where you'd find your Meissner's corpuscles. Here's your dermis, and the Pacinian corpuscles are found even kind of in the hypodermis as well. I mean, they're kind of staggered weird. So they're layered, and they're responsible for detecting deep pressure. Free nerve endings, right, can actually project up they're not protected by a sac, and they project up into the epidermis, and they're responsible for detecting pain and temperature. Now, glands that you need to know. Sebaceous glands secrete an oily substance called sebum that protects the hair. Eccrine sweat glands secrete a watery solution that cools you down, right? And apocrine sweat glands secrete a lipid-rich solution that you only find in the armpits and in the pubic hair, so your armpit hair and your pubic hair. And they play a role in mate selection. They also protect the skin from abrasion that you get in areas of the body that are kind of rubbing together. Sebaceous glands. So here we have a hair. Now, the part of the hair that comes out of the body that you can physically see is called the shaft of the hair. The part of the hair that extends into the integument is called the root of the hair. The hair itself exists within an epidermal derivative tissue called the hair follicle. So right over here you see histologically, this is a hair follicle with the hair missing. So we don't get into talking about the layers in this class of the hair follicle, but the hair follicle, we'll call it an epidermal derivative, meaning you see how the epidermis kind of folds in to form that hair follicle there. Now, the follicle surrounds and protects each hair, right? And associated with every hair follicle is a gland. And this gland, which kind of forms grape-like clusters, is called the sebaceous gland. Sebaceous glands secrete an oily substance called sebum, and that sebum travels through a duct where it's released into the hair follicle, and then that oily substance covers the shaft of your hair and protects your hair. It's kind of like conditioner. It's a natural conditioner, and it prevents hair from clumping too much, etc. So it protects the hair. It actually protects the hair. When you're looking here, this is your sebaceous gland. It secretes that oily sebum into the hair follicle where it ultimately coats and protects the hair. During puberty, things like hormones such as testosterone and estrogen can actually upregulate the activity of sebaceous glands. It can cause them to start cranking out more oil. So you'll notice that teenagers are pretty greasy or pretty oily. And it'll start cranking out more oil, right? And sometimes this duct where the oil gets released into the hair follicle can become clogged. And when it becomes clogged, that oil, that lipid-rich oil, is a great food for bacteria. So bacteria can colonize that area, and it can produce a little cyst that we think of as being a pimple. So, sebaceous glands. There's your sebaceous gland. Now you have two types of sweat glands. The more abundant type and smaller type of sweat glands are called eccrine sweat glands. Eccrine sweat glands produce a watery, electrolyte-rich fluid that's secreted, travels to a duct, and gets released onto the surface of the skin. That sweat is intended to evaporate and cool you off. It plays an important role in thermoregulation, can also play a role in detoxification because sometimes, right, things that our body is trying to desperately get rid of can get into the sweat and it can be eliminated. That's why if you've ever smelled somebody after a night of drinking, you can really smell them. Apocrine sweat glands, on the other hand, are only found in the armpits and the pubic hair or the groin. They're much larger, much more complex, and they secrete a lipid-rich or an oily solution that's always secreted into a hair follicle. And what it does, apocrine sweat, if you've ever noticed somebody exercise, if you look at the sweat stains in the armpits, they're not clear or white, they're actually yellow. And that's because apocrine sweat is very, very oily. And that apocrine sweat plays an important role in mate selection with things like pheromones that I'm not going to linger on too much. The apocrine sweat also protects areas that are subject to things like abrasion, right? It's kind of like body glide. Now, things that piss me off. Let's go ahead and look at the glands. So, sebaceous gland, Sebacean gland, eccrine sweat gland. You have a lot of them in the palms of the hands and the soles of the feet because those places get hot. Apocrine sweat gland. What's wrong with this? Well, the duct is not emptying into a hair follicle. So the duct should be emptying into a hair follicle, but you can see that it's much larger and you'd only find it in the groin and in the armpit hair. Now, what we're going to talk about, just really briefly, is hair. The only structures of the hair you need to know are the shaft of the hair follicle, the hair bulb at the very end here, and then the hair papillae, which is a network of blood vessels that actually feed into the areas of the hair that allow it to grow. And when you look at your labs, you can kind of go through that. Now, we have the nail. I'm not going to talk a lot about the nail. You can kind of look at the different structures of the nail that you need to know. It's an epidermal derivative. It protects the fingers and the toes, prevents distortion. It's a protective tissue. Plays a role in things like manual dexterity, how you grip things, etc. Can be used as a tool to like scratch. The hair matrix is where the new nail is growing from. You don't ever want to rip that. I had my thumbnail ripped out recently, and the only reason it grew back is because the nail matrix is because the nail matrix remained intact. We have the root of the nail. We have the cuticle, right? So go ahead and go through the different types of structures in the nails that you need to know. Then we have hairs. We have lanugo hair, which is the soft, fine hair that covers the fetus prior to birth. Sometimes when babies are preemies, they have this layer of hair on them. We have vellus hair, which is what we think of as peach fuzz. And then we have terminal hair, which is like the hair in our pubic area or on our head, etc. And that's that thick, pigmented hair found all over the place, kind of especially when we're adults. Now, when you think about hair, when I say epidermal derivative, so here we're talking about the hair follicle. And the hair follicle, right, when you break it down, consists of a connective tissue root sheath, a glassy membrane, an external epithelial root sheath, and an internal root sheath. So there are multiple layers to that hair follicle, but for this intro class, I'm just going to ask you about the follicle itself, but it's kind of important to highlight that. The hair root, which is the part of the hair that extends into the body, consists of the outermost portion of the root, which sits in the follicle, called the cuticle. So we often talk about the cuticle of the hair. The innermost portion, which consists of multiple cell layers, called the cortex, and then the deeper layer, and then the innermost layer called the medulla of the hair. The hair matrix is the cells in the hair that function very similarly to the stratum basale, where they're dividing and ultimately replacing the different cells in the hair that are coming up. And then the hair papilla provides the nutrients to that hair matrix so it can grow. And then you have your melanocytes cranking out the melanin if you have darker hair color. So when you think about that, I'm probably going to ask you questions on this model. And I'm going to say hair shaft, hair root, hair follicle, hair papilla, hair bulb, right? Now, when you do things like laser hair removal, What the the procedure is essentially they come down and they destroy this hair papilla with a laser. And if you destroy the blood vessel network that feeds to the hair matrix or the cells that allow hair to grow, that hair is never going to grow back. So you can only do that during the cycle of the hair in which the hair is actually growing, because the hairs go through different cycles. If you do it while the hair is resting and the hair isn't growing, you're not going to destroy that hair papilla and the hair is going to remain intact. So that's why you need to go through multiple cycles of that. Okay, y'all, have a good day. Bye-bye.